Hello everyone, welcome back to our channel. And today we'll be talking about such a technology which has not only revolutionized the field of medicine but also plant research and microbial genetics. Our topic is the CRISPR Cas system, which in the year 2015 was declared as the breakthrough of the year by the leading international scientific journal Science. Now, CRISPR-Cas, as you will see in the subsequent slide, is a bacterial acquired immune response. However, it has been harnessed as a powerful molecular biology tool by Emmanuel Charpentier and Jennifer A. Doudna, for which they won the Nobel Prize in Chemistry in the year 2020. They defined the CRISPR-Cas system as genetic scissors, a tool for rewriting the code of life. In today's video, we'll have a look into the origin and discovery of the CRISPR-Cas system and what are the steps in the bacterial adaptive immune response. And in the subsequent video, we'll discuss how this bacterial phenomena has been utilized as a precise genome editing tool. So let's start. Now, the history of CRISPR-Cas dates back to 1987 when a group of Japanese scientists were working with an E. coli gene alkaline phosphatase. So alkaline phosphatase is involved in the phosphate metabolism of the bacteria. Now they found that the genome of E. coli contains surprisingly repetitive DNA elements which were separated from one another by certain diverse or variable DNA sequence. So if you consider this to be the bacterial DNA, there are certain conserved repeat elements and they are separated by certain random DNA sequences. Now at that time, DNA sequencing was not that common. So this DNA module kind of remained a mystery. Now in the year 2002, the term CRISPR was coined. So CRISPR is an acronym which stands for Clustered Regularly Interspaced Short Palindromic Repeats. However, the function of this CRISPR locus was still uncharacterized. But a group of scientists, they showed that there are certain proteins called the CRISPR associated proteins or the Cas proteins which remain in close proximity to this CRISPR allele. So if I redraw the schematic, you have the Cas proteins, the repeat bacterial DNA sequences, and some random DNA sequences. Now, it was in the year 2005 when bioinformatic analysis was on a rise. A group of scientists led by Dr. Mojiko. So you can see that there were several scientists involved in the discovery of the CRISPR system. So Dr. Mojiko along with his colleagues found that these spacers or variable DNA sequences located within this repetitive DNA elements actually matched to the genome sequences of bacteriophages. So what are bacteriophages? These are viruses which infect the bacteria. So this provided with a preliminary hunch that probably the CRISPR-Cas array is involved in the defense response of the bacteria. Finally, the mechanism was dissected by Barango and his colleagues. So Dr. Barango was associated with a food processing company called Danisco. So basically, they were trying to increase the lifespan of Streptococcus thermophilus, which is a bacteria used for the manufacturing of yogurt or curd. And they found that when a bacteria is challenged with a virus, some segments of that viral DNA gets integrated into the bacterial genome in this CRISPR array. And if you remove or add new spacer element, the resistance of these Streptococcus strains to the phages could be manipulated. So they utilize this CRISPR-Cas adaptive immune mechanism to generate vaccinated Streptococcus thermophilus strain, thereby increasing the shelf life. So now let's have a look into the phases of the CRISPR-mediated immunity. So the first step is spacer acquisition, which is the integration of the viral DNA into the bacterial genome. Now this is mediated by a specific set of Cas proteins, Cas1 and Cas2. So basically, a part of the viral DNA is snipped off and introduced into the bacterial DNA by means of recombination. Now the segment of the viral DNA which is cleaved is known as the protospacer element. So the protospacer is associated with the virus. And when it gets integrated into the bacterial genome, it is recoined as the spacer element. So both this protospacer and spacer are identical in sequence, but it is referred to as protospacer in the virus and spacer in the bacteria. Now the next step is the transcription of this CRISPR locus. Now transcription leads to the formation of the CRISPR RNA or the cRNA. Now since there are several such random DNA sequences or spacer elements, it leads to the production of a long CRISPR RNA transcript, which is further processed by another set of Cas enzyme to produce the mature CRISPR RNA, which is around 20 to 24 nucleotide in length. Now these mature CRISPR RNA would further guide the Cas complex when a virus would infect the bacteria. So the final step is target interference. So when a particular bacteriophage reinfects this bacteria, the spacer element provides the danger signal that this bacteriophage is foreign in origin. So the transcription of the CRISPR locus and the processing of the mature CRISPR RNA takes place, followed by the association of an multiprotein effector complex composed of several subunits of Cas protein to assemble at the viral DNA and The nucleus activity of the Cas protein cleaves off this viral DNA, protecting the bacteria from the viral infection. So now, to get a better picture of this process, so you have this viral DNA infecting this bacteria. So this is the bacteriophage. A part of the viral DNA, which is the protospacer, gets integrated into the bacterial DNA as the spacer element. This is followed by the biogenesis of this CRISPR RNA. This is the precursor cRNA, which contains all the spacer element, which is further processed to 20 to 24 nucleotide long mature CRISPR RNA. And when this bacteriophage reinfects this bacteria, this spacer element helps in the recognition of the foreign mobile genetic element, association of the multi-protein Cas complex, and cleaving of the viral DNA, protecting or defending the bacteria from viral infection. Now, the CRISPR-Cas immune system has been broadly classified into two subtypes, class 1 and class 2, mainly depending upon the protein complexes which are associated with cRNA biogenesis and target interference. Now, Cas1 and Cas2, as you can see, these two proteins are common to all the classes and they are involved in the spacer acquisition. However, in case of class 1 CRISPR-Cas system, it involves a multi-protein complex which comes together for the biogenesis of the CRISPR RNA and the degradation of the viral DNA. On the other hand, the class 2 CRISPR-Cas system consists of a multi-domain single effector protein. For example, for class 2 type 2 it is Cas9, for type 5 it is Cas12, and for type 6 is Cas30, which performs both the cRNA biogenesis as well as the target interference. So as you can understand that the class 2 mode of CRISPR-Cas immune system is more amenable to genetic engineering owing to the presence of a single functional protein. And hence the CRISPR-Cas9 system has been widely utilized as the tool for targeted genome editing, which we would be discussing in our next lecture. Thank you.